Alright guys, so I am back with our part 2 of our review. We are going to talk about microscopes and cells, cell organelles, transport through the plasma membrane or cell membrane, and then photosynthesis, cell respiration, and ATP. Alright, so the microscope. So we have 3 specific microscopes that I want you to pay attention to and be familiar with. The first of which is the compound light microscope. Compound light microscope, those are the ones you're most familiar with. Those are the ones that you've used in school. So it's a typical light microscope. It is capable of magnifying things by 1,000 times. It's not great for seeing like details of the inner workings of a cell, or if we want to magnify something so we see like the hairs like we do on that little tiny insect on that next picture. But it does magnify things by 1,000 times, so we can see details from a perspective of cells dividing. We can see your hair shaft. You can see a bunch of things, like it's hard to see bacteria. They're very difficult to see. They're very small, so you've got to make sure you dye your slide and do some other things. But it does increase our ability. And like I said, that uses a light to shine up through the bottom of your specimen, so you need a nice thin specimen. But it is detailed enough for us to use in school and to magnify things greatly. The next one is a scanning electron microscope. Scanning electron microscope allows us to generate a 3D image of the outer surface of a specimen. It's really cool because you can magnify things by like millions of times. Like you can see grains of sand and specks of dust and like insects are really cool. So it uses electrons. It shoots these electrons at the outer surface of things, and it sends the picture back to a computer, and it's a computer-generated 3D image of the outer surface. And if you can remember, scanning because it scans the outer surface. Then we have the transmission electron microscope, and this we can see super detailed images of inside of cells, viruses, all sorts of really cool things. But we need a nice thin specimen because the transmission electron microscope is actually going to send electrons through the specimen. So that's why it's called transmission, because it's transmitting them through. But you can actually see the inside of things, which is really unique and cool. So we talked about the history of cells. We have these 5 scientists that we talked about before. We have Robert Hooke. Hooke looked at a piece of cork and is the one who's credited with discovering cells. So if you can remember, Robert Hooke discovered cells. He looked at a piece of cork and discovered cells. Then we have Leeuwenhoek. He looked at pond water and discovered little unicellular single-celled organisms swimming around in that water. Then we have Schleiden, Swan, and Virchow. And I've reviewed this several times with you guys. But Schleiden discovered that plants were made out of cells. And then Swan, kind of sounds like swan, discovered that animals are made out of cells. And then lastly, Virchow discovered cells come from other cells. They helped to come up with this thing that we call the cell theory. And we have 3 parts of the cell theory. We have the first part. All living things are composed or made up of 1 or more cells. Cells are the basic unit of structure and function of all living things of life. And all cells arise or come from pre-existing cells. Super important. Please memorize these. You will be asked this. Okay, then we talked about the types of cells. And if you remember, all cells, no matter what kind of cell you are, you will have DNA, cytoplasm, a cell membrane, and ribosomes. And we have 2 categories of cells. We have prokaryotes and eukaryotes. Remember, bacteria is the only thing that is a prokaryote. So prokaryotes are bacteria. Now remember, pro rhymes with no because prokaryotes have no nucleus and no membrane-bound organelles. They're very simple looking, okay? Very small, less complex looking than a eukaryote. They have a singular loop or single ring of DNA, okay? So that's a prokaryote. Pro rhymes with no because it has no nucleus and no membrane-bound organelles, and bacteria is an example. Then we have eukaryotes. Eukaryotes is everything else. So plants, animals, protists, and fungi are all examples of eukaryotes. U rhymes with do because they do have a nucleus and they do have membrane-bound organelles. They're much larger cells, much more complex looking. They have a lot of organelles, and they also have rod-shaped DNA. So if we look at these eukaryotic cell organelles, please review them. The important ones are the ones that are highlighted there. Really be familiar that the nucleus is where we can find the DNA. The mitochondria are like the powerhouse of the cell. This is where ATP is made. Remember, ATP is the energy molecule for our cells. Then we have the ribosomes. The ribosomes actually make proteins. You will probably be asked that. Then we have the chloroplast. And again, chloroplast is where photosynthesis in plant cells takes place. And then we have the cell wall, which is also in a plant cell that surrounds it. And if we compare plant cells to animal cells, remember I told you plant cells contain 3 organelles that animal cells do not. I said, remember the 3 C's. So a plant cell has a cell wall. It still has a cell membrane, but it has a cell wall that surrounds it. Okay, and then right inside there is the cell membrane. It also has chloroplasts because it does photosynthesis, and then it has this large central vacuole. Vacuoles store stuff for the cell. They store water and other nutrients. But a plant cell has a huge central vacuole for storing water because it can't just get up on its own legs and go get a glass of water. It needs a way of storing water. So when there's periods of no rain, unlike today with our So we need to talk a little bit about the cell membrane, the plasma membrane. In all cells, no matter what kind of cell you are, you have a cell membrane. The cell membrane is made of a phospholipid bilayer. Okay, phospholipids because it's got a phosphate, and then the little tails that are the lipids. Okay, we call the phospholipid bilayer for two layers, selectively permeable or semi-permeable. What that means is it allows some things to come through and keeps other things out. Scientists use this thing called the fluid mosaic model to describe the cell membrane because it kind of jiggles a little bit because it's made out of phospholipids. Lipids are fat, so they move a little bit. And then it's got a bunch of other little things, little protein channels and all sorts of stuff in those phospholipids that allow things to pass through. Now, things can pass through the cell via 2 ways, either passive transport or active transport. In passive transport, passive is just the word that it uses to describe it. When you're passive, you're just sitting back and letting things happen. That's exactly what the cell does. The cell does not use any energy, it just kind of sits back and lets things go back and forth across it. Okay? Then we have active transport. Active means it's actually got to do something, so it uses energy to actually move things back and forth. For passive transport, we have 3 kinds. We have diffusion, osmosis, and facilitated diffusion. So diffusion, small molecules just move back and forth with a concentration gradient. What that means is it moves from an area of high concentration, lots of particles, to an area of low concentration, okay, because it's trying to spread itself out. Osmosis, that's just a special word that we use for the diffusion of water. So osmosis is water moving across the membrane back and forth from areas of high concentration to areas of low concentration. Facilitated diffusion is diffusion, but it just needs a little bit of help. To facilitate means help. So actually, these are a little bit larger molecules that are going to still move with that concentration gradient back and forth, so areas of high concentration to low concentration. They're just going to use these things called protein channels inside that cell membrane. They're a little bit larger tunnels for them to pass through. Then we have active transport. Active transport involves the use of energy again. Okay, it's actively got to do something. So the first thing we have is endocytosis. Endo means to enter, and cyto means cell, so entering the cell. So large molecules are actually moved into the cell against the concentration gradient. So it's moving from an area of low concentration to high concentration. The cell actually grabs a hold of what it wants and surrounds it and pulls it inside. Okay. We have a specialized form of endocytosis called phagocytosis, which is used to fight disease. This is white blood cells that kind of like gobble up nasty stuff like viruses and bacteria and things that don't belong there and eat them up. Okay, so it's used to fight disease. Then we have exocytosis. Exo for exit, cyto for cell. So we have large molecules that are moving against the concentration gradient, so area of low concentration to areas of high concentration, and the cell actually has to take them and open up and push them out. Okay, so exocytosis uses energy as well. So when we talk about how osmosis actually works. So we can put a cell into 3 different kinds of solution. We can put it in an isotonic solution, a hypertonic solution, or a hypotonic solution. And we're going to talk about which way the water actually moves. So in an isotonic, iso for equal, because water is going to move back and forth across the cell membrane equally. So our cell is going to remain normal. This is like what we want. We want our cells to remain like this. Equal amounts of water going in and out. So everything's balanced. If you were to put a water or a cell into a hypertonic solution, hyper means a lot, like hyperactive, a lot of energy, right? So hypertonic has a lot of solutes outside of it. So the water is actually going to move out of the cell. So hypertonic makes the cell shrink down and shrivel up. Okay, we don't want that to happen. So what happens is we get really dehydrated. Okay, so water is actually moving out of the cell, making it shrink up really teeny tiny. And then we have hypotonic solution. If we were to put this cell into a hypotonic solution, if you remember, hypo has a big O because the cell is going to swell up big and fat. Water is actually going to move into the cell, kind of like a water balloon. And in animal cells, this is really bad because if our cells get into a hypotonic solution where there's just too much water going into our cells, the cell can actually explode. Now this won't happen for plant cells because they have a nice rigid cell wall that holds them and keeps them from absorbing too much water. But animal cells, they can, hypotonic solution, they can swell up big and fat like that water balloon that can actually explode. So now we're going to talk a little bit about ATP and ADP. So if you remember, ATP is the molecule that all cells use for energy, and ATP is the fully charged molecule, okay? So you actually take that molecule, ATP stands for adenosine triphosphate because it has 3 phosphate groups. You break apart one of those phosphates and you release energy. When you do that, it turns into ADP, adenosine diphosphate, di for 2 phosphates. You eat some food, recharge that molecule, you add a phosphate, and it turns it back into ATP, so it's a cycle. But our purpose is ATP is a fully charged molecule all cells use for energy. When we use up the energy, it turns into ADP, which is like a running low battery. We recharge it by eating food, turn it back into ATP. Okay, next thing we need to talk about is photosynthesis, and I know we've done a lot of review lately about that. But if you remember, photosynthesis is on the chloroplasts of plant cells. Leaf cells actually have the most chloroplasts because if you think about it, the leaves are actually nice and broad to actually help them absorb a lot of sun. Okay, so they have the most chloroplasts. And we use a green pigment chlorophyll to capture the sunlight. So we take an energy from the sunlight, and we use carbon dioxide and water to produce glucose and oxygen. And if you look down below, I've got my equation, and this is the balanced equation, so please remember this. So there's my sunlight, and there's my reactants. Reactants are what we're putting together to react. And in photosynthesis, my reactants are carbon dioxide, or 6 molecules of carbon dioxide, and 6 molecules of water. And they use the energy from the sun to make our products. And our products are glucose and 6 molecules of oxygen. Now we're taking in energy. The energy doesn't just disappear, it gets stored. It gets stored in that glucose molecule. Glucose is a sugar. And what happens when you eat sugar? You break it down and you release a lot of energy, right? So that energy gets stored in the form of glucose. Now we have cellular respiration. This is the opposite process. This is done in the mitochondria of both plant and animal cells. Yes, plants do photosynthesis. They make glucose and they make oxygen, but they need a way of breaking apart that oxygen to get the ATP energy. So they also do cell respiration. So cell respiration breaks down glucose. That's called glycolysis. That's the breakdown of glucose. And oxygen to produce carbon dioxide and water. And so we have our chemical equation for cell respiration. We've got glucose plus 6 molecules of oxygen, and those are our reactants. They're going to react to form our products. Our products are carbon dioxide, or 6 molecules of carbon dioxide, and 6 molecules of water. We do this, we break apart that glucose, and we release energy in the form of ATP. So the last thing I need to talk about is fermentation. Now, normally cell respiration is a process known as aerobic. It requires oxygen, right? We take in glucose and oxygen to break apart that sugar. We do glycolysis. We break it apart to get that ATP. Well, sometimes that is not, oxygen is not readily available. So we have this process called fermentation. Okay, so fermentation allows glycolysis to break down the glucose to continue without using oxygen. We call it anaerobic, without air, to keep making ATP. Now we use this when we make some types of food like cheese, yogurt, bread. All these foods, we add some kind of bacteria or fungus like yeast to break down the sugars found in these substances, and they do this in an anaerobic process, fermentation, without the use of oxygen. Okay, this is also used when you work out super hard and you can't keep up with the amount of oxygen required. At some point, your muscle starts to get fatigued or tired, right? And you're breathing super hard. You're still trying to keep up with forming all this ATP for your cells to use for energy. So your body switches into fermentation mode. And in fermentation, it's not using all of that oxygen. It's making ATP without using the oxygen. So it builds up this other chemical called lactic acid. And lactic acid is what causes those muscles to cramp. Okay? So anyway, we've talked about a whole heck of a lot here. Please review it. Review your notes. I have added some review questions, so you can find these on the actual PowerPoint. Plus, make sure you go back and answer discussion questions to help you review. I'm giving you points for those. If you need anything, you have any questions for me, please, by all means, ask me. We are still planning to do our benchmark exam on Monday and Tuesday this week. Those of you that are e-learning students, yours will be scheduled for Wednesday, and you'll receive an email from me with the time and with directions on how to log in, because I will be monitoring you for that benchmark exam on Microsoft Teams. Okay, so again, if you have any questions or you need anything, please, by all means, ask me. Please review these questions, see if you can answer them, review the PowerPoint. Not just these videos, these questions are attached to that PowerPoint as well, so you've got plenty of time to look them over. All right, guys, and I will talk to you soon.